Our next section of Chapter 16 deals with imine and enamine formation reactions. Let's start with imines. So imines are compounds that contain carbon-nitrogen double bonds. They can have hydrogen or other alkyl groups attached here, and they can be derived from aldehydes or ketones. Historically, and you'll encounter this terminology in biochemistry, these are called Schiff bases. They're basic by virtue of that lone pair of electrons on nitrogen. The reactions occur by the formation of the amine occurs by the reaction of an amine, primary amine. Notice there's only one alkyl group here, with an aldehyde or ketone, typically in the presence of an acid catalyst. That amine can be ammonia. Simple primary amines, aniline will suffice. Methylamine is shown in the example here. The end result is the imine, the carbon-nitrogen double bond compound, containing the alkyl group, if there's one attached to the amine. The byproduct of the reaction, like acetal formation, is water. So like we did with acetal formation, let's jump right into understanding what the mechanism of the reaction is, and we'll work our way through that. So we're going to use as our example the reaction of methylamine with acetone and an acid catalyst. And the acid catalyst in these reactions is often, as it is in the acetal reaction, something like para-toluene sulfonic acid. These reactions may or may not take place in aqueous medium. So we're going to start with acetone. And most steps in the mechanism are equilibria steps. In methylamine, amines are quite nucleophilic, and so no catalyst or anything is really necessary in order for the reaction to begin. Even the weakly electrophilic acetone will react with methylamine to give an adduct. So it's a typical nucleophilic addition to a carbonyl compound. In this case, it's going to give us this protonated ammonium ion as an intermediate. We'll assume this reaction is taking place in water as a solvent. Water is often used in these reactions because it's both a little bit basic and a little bit acidic. And so we can deprotonate this species with water. Oops, I'm missing a hydrogen here. Produce this alkoxide intermediate, which is then protonated. So the water here is kind of serving as a proton carrier, if you will. Which brings us to this species, which is analogous to a hemiacetal. It's an aminal. Oops, aminol. Remember, we have an acid catalyst present, so there's going to be some hydronium ion. The next thing that happens is we protonate the hydroxyl group. Again, this is analogous to what we saw in the formation of an acetal. Just like in formation of an acetal, we get elimination. Of the water. But in this case, the final step is deprotonation of this species. Regenerates the catalyst and forms the imine. So there's our mechanism of the formation of an imine. So this reaction is widely applicable, and if you look back in history literature, you find many, many, many examples of people forming imines of one sort or another from aldehydes and ketones. Turns out this is really quite useful in terms of characterizing low molecular weight liquid aldehydes and ketones. Often the imines were solids that could be easily characterized. You can look up in databases melting points of derivatives of aldehydes and ketones made from various amines as derivatives. So many of these have properties that make them likely to be solids. So for instance, sebicarbozones are often formed. This collection of very polar atoms makes the compound very polar and prone to crystallizing, for instance. There are other examples. Before I go to the next slide, notice here that in some of these reagents, particularly this last reagent, we have an NH2 group here, we have an NH2 group over here. The one that's next to the carbonyl is not particularly nucleophilic. It's this terminal NH2 group that reacts. Notice this is the nitrogen here, which corresponds to that one. Here are some other examples of derivatives of hydrazine. So the hydrazine part of the molecule is here. This is 2,4-dinitrophenylhydrazine. I've mentioned that compound to you before. It's a bright orange compound, and the derivatives are bright orange. They also tend to be crystalline solids because of the high molecular weight and polarity. This is tosylhydrazine. Here's that tosyl group, the para-toluene sulfonyl group. And these also tend to be nice crystalline solids. And you can even use something as simple as hydroxylamine, and these form these oximes, which have a lot of synthetic uses. But in each case, the active part of these molecules is the NH2 group. As is the case with the previous reactions, formation is reversible. So in most imines can be hydrolyzed right back into the amine that you started with. And like acetal formation, this really has to do with the amount of water in the mixture. So if you use an excess of water in an acid catalyst, you can reverse this reaction. Now, if you haven't already anticipated it, you know what I'm going to ask you to do. Rate the mechanism. You should be able to work your way backwards in that reaction. One of the reasons that imines are interesting is that they show up in biological systems in a variety of different ways. One key way that imines show up is the way that cofactors are incorporated into enzymes. So you're all familiar with the adage that you should, make sure that you eat all your vitamins and those sorts of things. And one of the reasons that you need to do so is many of the things that we call vitamins, things like vitamin B6, are essential for certain enzymes to function. And it happens that pyridoxamine phosphate, which is vitamin B6, actually is a cofactor that's required for the formation or the synthesis of many amino acids, particularly the amino acid aniline. In the way it functions, it sits inside of an enzyme active site, and its active part is this amino group. And its role in those biological systems is to react with this ketone, this is called pyruvate, and it forms this imine intermediate. And the key thing that's happening here is the nitrogen is being added to this carbon. You have a carboxyl group adjacent to it. When this group is ultimately processed, it becomes the amino acid aniline. And so there's an imine formation in a biological system. Let me give you another example of an important imine formation of biological system. And this also is related to one of those old adages. You probably know that you should eat your carrots because they're good for your eyes. Well, I'm going to tell you why. So carrots and other red vegetables, red peppers, tomatoes, those sorts of things, contain pigments. And one of the most important pigments, at least from a dietary point of view, is this big molecule. This is beta-carotene, and you can tell where it got its name from. It's a long conjugated molecule, and that conjugation results in the molecule absorbing light in the visible region and appearing orange or red. When we eat carrots or tomatoes or tomato paste and those sorts of things and consume this, our body processes it into a compound that's essential for our eyes to function. The first step in the metabolism of this when we eat it is it gets oxidatively cleaved. This carbon-carbon double bond is cleaved by this enzyme, beta-carotene 15,15' dioxygenase, and turns it into this. This is called retinol, or it's actually known better as vitamin A. And they add this to foods that are supplemented with certain vitamins. That is then further processed by our bodies by an oxidation reaction, converting it to an aldehyde, which is known as retinal. Retinal is isomerized by another enzyme called retinal isomerase. And it has this curious thing that it does is it very, very specifically converts this double bond in the molecule from its trans configuration to its cis configuration. And in this form, this molecule now is essential for vision. So let me explain how that works. So our bodies, after we eat our carrots, process the beta-carotene by that oxidative cleavage, oxidation, and isomerization into retinal. Retinal is then incorporated into a protein. That protein is called opsin. So where it says the word protein here, it has inside of the protein, there's an amino group. That amino group couples with this aldehyde to form an imine, and this combination is called rhodopsin, and it sits in the retina of our eyes. This molecule and that protein are light sensitive. And when light, a photon of light, interacts with that protein retinal imine complex, it causes that double bond to isomerize back into the trans. And that change in shape triggers a neurosignal. So the rhodopsin absorbs light, causing we call this a photoisomerization because it's the light that's causing it to happen, and that triggers a nerve impulse. The nerve impulse is actually amplified, ultimately reaches our brain, and is interpreted as a spot of light in some way in the back of our eye. In this form, it's no longer photosensitive, and so it has to be reprocessed. And so our bodies then kind of recycle it. They hydrolyze the imine bond. I said imine bonds reactions are reversible. The now all trans retinal is released, and then it's recycled through that same pathway where it's reisomerized into the cis form, reincorporated into the opsin to make rhodopsin. The other thing that this allows our body to do, the fact that there's this recycling process, is it allows us to create more or less rhodopsin depending upon the ambient light conditions. So if there's very little light available, our eyes will make more and more rhodopsin in order to make them more sensitive to the low levels of light. On the other hand, if we go from a low light situation into a high light situation, walking from indoors to outdoors, for instance, our bodies will hydrolyze the rhodopsin and decrease our eye sensitivity. So you've probably noticed this when you walk from a dark indoors outdoors. It takes a few moments for your eyes to adjust. And that adjustment is basically these reactions occurring in one direction or another, either breaking down some rhodopsin to decrease our sensitivity or creating more rhodopsin to increase our sensitivity. So there's chemistry that has to happen. It doesn't happen instantaneously. So understanding this whole process is actually work that was awarded the Nobel Prize in Physiology and Medicine. So this is back in 1967. The award was shared by Ragnar Granit, Haldan Hartline, and George Wald. And you can see that the Nobel Committee awarded them here for their discoveries concerning the primary physiological and chemical visual processes in the eye. They fleshed out through a series of experiments how this whole cycle worked in different ways. So those reactions that form the imine were reactions of primary amines with aldehydes and ketones. What happens if you have a secondary amine? So this is a secondary amine. Notice the nitrogen has two alkyl groups attached to it. Here we have an aldehyde or a ketone. You'll notice I've drawn a little bit more out here, and that's important. When that reaction occurs, you don't get an imine. You actually get this kind of compound. It's called an enamine, ene for the double bond, and then we have an ordinary amine, the nitrogen with 3 single bonds to it. So here's a couple simple examples. Cyclohexanone here reacting with pyrrolidine, and we form this pyrrolidine enamine. Here we have dimethylamine reacting with acetaldehyde, or excuse me, that's acetophenone, and we get this enamine that you see over here. All right, so like we did with the others, let's mechanistically figure out how this works. So the overall reaction is shown here. We need amine, we need an aldehyde or a ketone, we need an acid catalyst, and these are our products. We form the enamine, and like our other reactions we've seen to this point, we have water as a byproduct. So let's draw out the mechanism. So the reaction begins like the imine formation. Remember, amines are good nucleophiles, so we don't really need any catalyst at this point. The catalyst plays another role. We get nucleophilic addition, forms an alkoxide intermediate, this ammonium ion. This is deprotonated. And then protonated. And we form our aminal. And again, continuing on in the same vein as imine formation, we're going to protonate that hydroxyl group. And that's the key role for the catalyst here. It's converting that hydroxyl group into a good leaving group, and it undergoes elimination like we saw in the previous mechanism, like so. So we're going to form water. But very importantly, we get to here. And now, because we have two alkyl groups, we can't deprotonate the nitrogen. So the only way this compound can neutralize is to lose a proton, and it loses a proton here, alpha, to the nitrogen. So this is our reference position. This is the alpha carbon, and so this is called the alpha hydrogen. And so we end with the deprotonation step, but the deprotonation step occurs there, and we end up with an enamine. So there's our enamine, and again, we can account for the fact that we've regenerated our catalyst. So there's our look at the formation of imines and enamines.